Good afternoon, good morning, good evening. I am Anne Gourmelon, Principal Administrator of the OECD Test Guidelines Program. It is my pleasure to welcome you to the OECD webinar on the implementation of the defined approaches on skin sensitization for chemical safety in OECD Guideline 497, published in June this year. So we will start the webinar with a joint presentation from Silvia Cassati and Nicole Klein-Stroyer. Next. Dr. Silvia Cassati is a Senior Scientific Officer at the European Commission's Joint Research Center in Ispra in Italy. She has been coordinating the activities of the JRC related to the evaluation of non-animal test methods for skin sensitization in support of their regulatory acceptance. Dr. Nicole Klein-Stroyer is the acting director of the NTP Interagency Center for the Evaluation of Alternative Toxicological Methods, NICETM, which is the US federal resource for alternatives to animal testing. She was the US lead on the OECD project to develop the DHs guideline. Following a short Q&A session, we will hear a presentation from Laura Rossi on the use of the DHs under REACH. Dr. Laura Rossi is a scientific officer at the European Chemicals Agency. Her work includes assessment of REACH registration dossiers with the expertise in sensitization and general immunotoxicity. She's also a member of the OECD expert groups in relation to skin sensitization. Then, following again a short Q&A session, we will hear perspectives from two panelists, Paul Brown from US FDA and Anna Lowitt from US EPA. Dr. Paul Brown is an associate director for pharmacology and toxicology in the Center for Drug Evaluation and Research at FDA. His experience includes work on drug evaluation. He has participated in several OECD expert groups, including the defined approach on skin sensitization expert group. Dr. Anna Lowitt is currently the Senior Science Advisor at the EPA's Office of Pesticides Program, where she advises senior managers and she leads multidisciplinary teams on a variety of cross-cutting topics related to human health and ecological risk assessment. Some of these topics include new approach methods. She's currently one of the co-chairs of the Interagency Coordinating Committee on the Validation of Alternative Methods, ICVAM. And our last presentation today will be from André Mueller on the work to introduce non-animal methods under the UN Globally Harmonized System for Classification and Labeling. Dr. André Mueller works as a regulatory toxicologist at the Dutch National Institute for Public Health and the Environment. He's one of the leads of the GHS working group on the introduction of non-animal test methods into the GHS criteria. After that, we will have a final Q&A session. And if you just click, okay, I would also like to acknowledge my colleague Patience Brown from the OECD Secretariat, who has been instrumental in developing this OECD guideline. Thank you for your interest and enjoy the webinar today.
Good morning, everyone, and thank you, Anne, for this nice introduction. What Nicole and I will attempt to do with this first part of the webinar is to guide you through the main features of OECD Guideline 497. I will start by providing a little bit of background on the need for this guideline. I will then touch on the work that was conducted to increase the confidence in the defined approaches, with a specific focus on the LLNA and human reference data review. And then I will introduce the defined approaches currently covered by the guideline. Nicole will talk us about how uncertainties have been characterized and addressed. She will explain the performance of these DAs and will talk us about future activities and plans. Going back to the need for this guideline, I believe that you are well aware of the progress that was made over the last years in the validation and regulatory adoption in the area of skin sensitization. And we are talking about non-animal methods, which are addressing the first three key events of the skin sensitization adverse outcome pathway. Because of the limited mechanistic coverage of these test methods, none of them can be regarded as a full replacement of the animal tests. Actually, if we look into individual test guidelines, we can see that they prescribe these methods should be used in combination with additional data, with other evidence, specifically to conclude on the absence of skin sensitization potential, or to provide information that can satisfy the need for potency subcategorization according to the GHS. For these purposes, it is suggested to use these methods in the context of a weight of evidence approach or integrated approaches to testing and assessment. These are nevertheless quite flexible. So we were talking about the use of the test methods in isolation and the instructions to use data from these test methods in the context of integrated approaches to testing and assessment, which are quite flexible in nature, and therefore they do not guarantee a harmonized use and acceptance of the in vitro data. If you could go to the next one, please. In contrast to IATA, defined approaches use a defined set of information sources. So basically, the type of test methods that are used within are predefined. There is no choice by the final user on which are the inputs to the defined approaches. And also the data interpretation procedure is fixed. And therefore, defined approaches remove any kind of expert judgment. They are not flexible, and they are suitable for harmonization. The work that was done on the development of Guideline 429, if you could go to the next one, please, builds on previous evaluations on the defined approaches. These have been extensively evaluated in the context of activities within Cosmetics Europe, and also reported in a harmonized way in the OECD Guidance Document 256. The defined approaches were already agreed that they would have satisfied the majority of regulatory needs, especially in terms of classification schemes. Therefore, the project was included in the OECD Test Guideline Program in 2017 and was led by the US, by the European Commission Joint Research Center and Health Canada, and was conducted with the support of the expert group on defined approaches for skin sensitization. That was composed of scientific experts from regulatory agencies, validation bodies, and non-governmental organizations and industries. It was a huge group that really did a lot of analysis to bring this guideline to adoption. Next slide. The defined approaches included in the guideline have been assessed using an evaluation framework that was developed upon request by the OECD WNT and was subsequently agreed upon by the expert group. The elements that were considered for the evaluation of the defined approaches included, in first place, the information provided by the defined approaches. Whether this would be it would allow an equivalent regulatory use as the animal test. The mechanistic and biological relevance with respect to the existing sensitization AOP framework, the assessment of the predictive capacity that was expected to be equivalent or better than the one of the animal test to predict the responses in human, the reproducibility of the defined approaches, the characterization of the applicability in terms of technical limitations, but also in terms of chemical space tested, the fact that the data interpretation procedure could be easily understood and applied by the end users, and of course the transparency of the information, that all the elements necessary for review would become available to allow an independent evaluation by third parties. Next slide. Now let's see how the guideline is structured. The guideline is divided in sections. In section 1, you will find a general introduction, the DA's proposed use and use scenarios, and the limitation of the DAs. Section 2 of the guideline include the DAs which are proposed for hazard identification. Section 3, the DAs proposed for potency categorization. At the end of the guideline, there are a series of annexes and appendixes that provide additional information on how to apply the DAs and how to interpret the prediction. In addition, all the analyses that were performed in support to the development of this test guideline can be found in the supporting document that is published as a series on testing and assessment number 336. Next slide. The overall purpose of the guideline is to provide information that is equivalent to that which is provided by the standard animal test, the local lymph node assay. Of course, this is to reduce reliance on animal testing as requested by the different regulatory settings and as embraced by the OECD. It's important that we keep in mind that as for any test guideline, especially for the purposes of classification and risk assessment, the outcome predictions from these DAs should be considered together with all available information existing on the test chemical. This is the first guideline which falls under the mutual acceptance of data of the OECD, the first guideline on defined approaches. And this simply means that the countries cannot require registrant to rerun the same defined approaches or test. The mutual acceptance of data does not mean automatic classification risk assessment, since this is under the responsibility of the specific regulatory jurisdictions and authorities. Next one. Now, going back to the work that was done to increase the confidence in these DAs, a lot of resources were put in the review of the in vivo reference data, the LLNA and the human data. The predictive capacity of the DAs was then evaluated using this highly curated reference data. We were in an unprecedented situation of having both the LLNA and the human data. And in relation to the LLNA, this has been reviewed in relation to the LLNA data were reviewed. Each individual study was analyzed against criteria that were agreed upon by the expert group and that were based on the essential test method components of the local lymph node assay performance standards, and also on a number of other considerations, such as, for example, the availability of the concentration tested and the corresponding stimulation indexes values measured. Furthermore, the expert group developed criteria and applied them consistently to all the LLNA studies with an estimated concentration 3 outside the measured dose range, so the so-called extrapolated AC3, to make sure that the estimated EC3 was sufficiently reliable to be used for the purposes for potency subcategorization. Particular scrutiny was also applied to the negative studies that were accepted only if all test concentrations, the stimulation indexes for all test concentrations were below 3, and the highest concentration used was at least 50%, unless scientific and sound scientific justification was provided. For evaluating the performance of the DAs for GHS classifications, at the end we came up with 156 test chemicals that were used, because for 12 chemicals it was not possible to assign with sufficient confidence the potency subcategorization on the basis of the LLNA data. Nevertheless, these 12 chemicals were used to evaluate the DA's performance for hazard identification. Next slide, please, Nicole. Similarly to what was done for the LLNA, an in-depth analysis was conducted on the human predictive patch test data, which included an evaluation of the source of variability and uncertainty in the data. In addition, a decision logic was developed for classifying the substances for their skin sensitization potency according to the GHS. And this was consistently applied to all the studies, including considerations of the borderline results. The review of the human data resulted in 63 chemicals with reliable GHS potency categorization, and three test chemicals that were only used for assessing the performance for hazard identification, because once again, for these 3 chemicals, a GHS subcategory could not be assigned with sufficient confidence. Next slide, please. Now we come to the DAs, which are currently included in Guideline 497. All the DAs which are currently included are based on the use of validated and OECD adopted test methods: the direct peptide reactivity assay, the keratinosense, and the HGLAT. For these methods, transferability within and between laboratory reproducibility have been characterized in phase of validation. The first defined approach, which is described, is the 2 out of 3 defined approach, which is proposed for hazard identification. Basically, this is based on, it's covering at least 2 of the first 3 key events of the adverse outcome pathway. The test methods are run in an undefined order. If the first 2 assays provide a consistent result, then the chemical is predicted accordingly as a sensitizer or non-sensitizer. Instead, if the first 2 assays provide a discordant result, the 3rd assay is run for the 3rd remaining key event, and the overall final conclusion is based on 2 concordant results, taking into account the confidence of the prediction. Next slide, please. The second part of the guideline includes DAs that, in addition to hazard identification, provides also information on potency categorization. Currently, there are 2 of these DAs described in the guideline: the ITS version 1 and the ITS version 2. The ITS DAs uses test methods that address key event 1, the DPRA, and key event 3, the HGLAT, in the adverse outcome pathway, and also include an in silico prediction that is provided by direct nexus in the ITS version 1 and by the OECD toolbox in the ITS version 2. The data interpretation procedure in these DAs involves that a score is assigned to the quantitative readouts of the DPRA, the H-CLAP, and the in silico prediction. And by summing the scores, a total battery score is derived. If this total battery score is equal or greater than 2, then the chemical is classified as sensitizer. Whereas if it is less than 2, the chemical is not classified. For the purposes of potency categorization, a total battery score of 6 to 7 identifies the strongest sensitizers, the GHS 1A class, and a total battery score of 2 to 5 identifies the weak to moderate sensitizers, covered by the GHS under class 1B. This was my last slide. I will then pass over to Nicole for the second part of the presentation.
Thank you, Sylvia. So again, apologies everyone for the technological difficulties. So I'm going to now move to the next half of the presentation. So I think as everyone is aware, this was really a landmark moment in the development of products for the OECD. So this is the first defined approach guideline that is intended as a full replacement for an animal method. Hopefully it's not the last, but there are several things that make this guideline unique. So within the DHs guideline, we have things like an approach for defining the applicability domain of the defined approaches, and that includes the applicability domain of each of the information sources, so both in vitro and in silico. We have decision trees for each defined approach that characterizes the uncertainty within those information sources and gives you a degree of confidence in the overall prediction, and I will walk through those for you in subsequent slides. We have approaches for both standardizing and reporting in silico predictions, because this, again, is the first time that an in silico information source has been included in a OECD guideline. And then we have also within the discussion in the introduction, and then some of the work that I'm going to talk about that we're doing to follow up, throughout the course of the project, we recognize that there are areas where additional research may be needed because there are certain chemistries where the in vitro approaches, the defined approaches, disagree with the animal models, but actually agree with the human response. And so there's a fair amount of evidence that the existing animal tests may not be predicting the human response well for particular types of chemistries. And so that's something that we are currently looking into a little bit more to try and further establish scientific confidence in the in vitro human mechanistic based defined approaches. So Sylvia already told you about the 2 out of 3. This is the confidence workflow. So this is a decision tree for the 2 out of 3 defined approach. So as she already said, you go through and you just conduct 2 assays. It doesn't have to be the first 2 key events. It can be any 2 out of the 3 key events. It's important to note that within the guideline, the current test methods that are included as information sources are the DPRA, the keratinescence, and the HCLAT, but that may be expanded in the future. But each of these decision points is where we take into account limitations from the individual key event-based test guidelines. So within each of the test guidelines for each of those methods, there are limitations that are described. So one example is for log P, so chemicals with a log P greater than 3.5. Within TG 442E, it says that negative results for the HCLAT are not considered reliable for those high log P substances. So that is a limitation that is carried over into the defined approach. So that's important to note. But then there's also this concept of borderline results. And this is something else that was developed throughout the course of the defined approach project. So the uncertainties here are actually captured by deriving statistical borderline ranges for each of the in vitro methods. And I'm going to walk you through what that looks like in the next couple slides. But one final thing that I want to note on this is that even if you derive an inconclusive result from the outcome of a defined approach, those results may still be used in the context of weight of evidence and IATA, integrated approaches to testing and assessment. So what do the borderline ranges look like? This is from Annex 1 to Guideline 497. This is a flowchart of the direct peptide reactivity assay prediction model. So this is looking at the mean depletion between cysteine and lysine, and this is taking into account borderline ranges and multiple runs to conclude on borderline results within the 2 out of 3 defined approach. So here, the original threshold for classification was 6.38% depletion, mean depletion. And then there was a statistically derived borderline range around that threshold, so between 4.95 and 8.32%. And so if you have depletion within that borderline range, then you need to follow this flow chart and you need to conduct a second run. It's also important to note that in the case of coelution with the lysine peptide, there is an equivalent flow chart to apply to the cysteine-only prediction model. Similarly, in Annex 1, we have a flow chart for the keratinescence prediction model, also taking into account borderline ranges and multiple runs to conclude on borderline results within the 2 out of 3 defined approach. So here, the original threshold for a positive classification was 1.5-fold induction, and then the statistically derived borderline range around this threshold was 1.35 to 1.67. So anything below that is considered negative, anything above that is considered positive, but within that borderline range, you need to perform another independent repetition and then conclude on that based on the decision logic here. And same for the HCLAT. The HCLAT also has borderline ranges that were statistically derived. So again, the original threshold was 150% induction of CD86, and that borderline range is between 122 and 184%, and 200% induction of CD54, and that borderline range is 157 to 255. So again, these very clear and easy to follow flowcharts will allow you to decide whether or not your test result is borderline and whether you need to perform an additional independent run. So the expectation for both keratinescence and HCLAT is that 2 independent runs are performed to conclude on whether or not a substance is a final outcome of positive, negative, or borderline. So these considerations are also taken into account in the integrated testing strategy defined approach. So again, here we have the limitations from the individual test guidelines, such as the log P limitation for negative HCLAT results. And then we also have a decision point where we take into account the applicability domain of the in silico information sources. And I'll talk through those in the next couple slides. But here at the bottom, you have the different combined scores and the outcome, in this case, for potency subcategorization. And then you have areas where you may derive an inconclusive result. And again, those can still be used in the context of weight of evidence and IATA. So what do the applicability domains look like? These are also included in the Guideline 497. So this is from Annex 2 of the guideline. This is Annex 2 includes both the protocol for running Derek Nexus as an in silico prediction model, as well as the definition of the applicability domain. Again, a very straightforward decision tree. All the positive predictions are considered in domain. The negative predictions are considered in or out of domain based on the presence of misclassified or unclassified features. Similarly, the protocol for running the OECD QSAR toolbox automated workflow is included in Annex 2, and also this consideration of the different applicability domain layers for the QSAR toolbox predictions. There are 3 layers here. So the structural layer is defined based on the atom-centered fragments derived from the structural characteristics of the training set substances. The parametric layer is dependent on 4 physicochemical properties, and then the mechanistic layer is determined by the predicted capability of a substance to interact with skin proteins without or after abiotic activation being taken into consideration. And again, for both of these information sources, many more details are contained in Annex 2 of Guideline 497. So now getting to sort of the punchline here. This is the first table in the guideline, and this shows you the performance of the defined approaches against both the LLNA reference data that Sylvia told you about and the human reference data that she described as well. So again, here we have around 168 reference substances for the LLNA and around 63 or so substances for human. There are slight differences by one or two chemical in each of these comparisons just based on the availability of the input data, but they're all pretty close to these numbers in terms of their basis for comparison. And so you can see if you're just comparing to the animal reference test, we get robust performance of each of the three defined approaches against the LLNA reference data, both for hazard and for potency prediction. And then importantly, when we compare to the human reference data, we really do much better than the LLNA. So here you can see in these columns, this is the hazard performance versus the human reference data for each of the three defined approaches as compared to the LLNA performance against the human reference data. And then in the last column, you see the GHS potency performance for both of the ITS defined approaches in terms of a class-wise predictive performance compared to the LLNA. And in every case, the defined approaches are equivalent or better than the LLNA in predicting that human reference endpoint. So where do we go from here? So some work that's been ongoing over the last couple of years that's being led by Dori Germelek at the U.S. National Toxicology Program is to try and understand and better characterize the applicability domain of the defined approaches that are based on the in vitro and in chemical test methods. So we polled our US federal and international regulatory partners for nominations of different types of chemicals that we could test in the deep red, the keratinocytes, and the H-clat. So those included things like formulation products and typically challenging chemicals and in vitro systems to try and understand how well these approaches may do against those types of chemicals or those types of substances. So that's ongoing. The testing of around 200 substances is largely done, and we're just analyzing the data now. I think that it's important to note that now that this guideline has been developed and approved and published by the OECD, defined approach developers really now have all of the information they need to demonstrate equivalent performance to the DAs that are covered by this guideline for hazard and potency characterization. To that point, there are two proposals under development for hopefully for review this cycle by the OECD WNT. So we're looking at developing sort of a Me Too DAs proposal where we look at the information sources that are currently included in the key event-based test guidelines. So 442C, 442D, and 442E. So those are things like the ADRA or the LUCENS or the USENS. And if we can substitute those information sources within the existing defined approaches that are already in Guideline 497. So that's one of the proposals that's under development. And then the second proposal is to expand the guideline and include a defined approach that provides a point of departure for quantitative risk assessment. Because as Sylvia mentioned, the current defined approaches are simply for hazard and potency categorization. And so there are certain regulatory authorities throughout the world that do quantitative risk assessment based on skin sensitization as an endpoint, and so would need defined approaches that can help them with that decision context. So in the last minute or two, I'm just going to talk you through just a couple of pieces of progress from the US regulatory side that my colleagues on the panel can expand upon further. So as far as the US FDA is concerned, last year, they issued draft guidance on non-clinical safety evaluation of the immunotoxic potential of drugs and biologics. So they are no longer recommending the LLNA. And within this guidance, they state that they will consider a battery of in silico, in chemical, and in vitro studies that have been shown to adequately predict human skin sensitization with an accuracy similar to the existing in vivo methods. So I think it's fair to say that the defined approaches within Guideline 497 fit exactly that definition that's within that guidance. The US EPA in 2018 had actually already released a draft science policy on defined approaches that they will accept as alternatives to the LLNA. So they have the 2 out of 3, and then they also have a sequential testing strategy that's based on tests for key event 3 and key event 1. And both of those DAs within that science policy already cover those Me Too information sources within each of those respective key event-based test guidelines. They have also really pushed the envelope as far as risk assessment is concerned and applied quite a reliable defined approach that's been peer-reviewed and published and reviewed by the ICFAM Skin Sensitization Expert Group, which was an artificial neural network model developed by Shiseido to derive essentially an EC3 equivalent based on the in vitro information sources. And they used that information on a group of isothiazolinone chemicals in their hazard characterization document in support of the FIFA registration review. And they use this to derive the point of departure for doing the quantitative risk assessment for skin sensitization of these compounds. And this is open for public comment at the moment. And then finally, we're very excited to announce that Nicedum has entered into an agreement with Unilever to collaboratively test and further develop their skin allergy risk assessment, or their SARA predictive model. So this uses a variety of input data to estimate a probability that a chemical will cause an allergic skin reaction in humans. And the intention here is to evaluate and test the SARA model using a variety of chemical data sets, including chemicals of interest to US and international regulatory agencies, and to make this model openly available for public use via the Integrated Chemical Environment Nicedum dashboard. So just to close, I think there were a lot of lessons learned in this project. Those of you familiar with all of the work that went into it know that it was over a decade, actually a couple decades of work in terms of developing the in vitro methods and conceptualizing of the defined approaches, evaluating them under the Cosmetics Europe collaboration, and then that was all prior to this project being included on the OECD work plan in 2017. And so here we are 4 years later that we finally have this new guideline. So it was a really incredible learning experience for all of us. But I think it's important to recognize that things like having reliable reference human data is a luxury that we don't have for other endpoints. And so as we evolve towards developing more defined approach guidelines, I think it's really important to shift our focus a little bit more to the human, biological, and mechanistic relevance of the defined approach. Obviously, we need to consider the animal reference standard, but it can't be the only basis for comparison, in my opinion. And I think that also as we evolve into further iterations of this guideline, and as we work on other guidelines, there really strong consideration should be placed towards recognizing the value of combining these information sources to overcome the individual limitations of the individual methods, because that's really what defined approaches are intended to do. So there were a huge number of people that contributed to this. I've probably forgotten some specific names, but some of the folks that really worked hard on this are called out. And then, of course, all of the members of the expert group on defined approaches for skin sensitization, around 70 of them, were absolutely instrumental in this process, and it could not have happened without them. So thank you very much, and I will stop sharing.
Thank you so much, Nicole and Sylvia, for this joint presentation. We will take 1 or 2 questions live, And our panelists today will also type in some answers for questions that we can't take live. So as we progress through the webinar, we will have more panelists to whom the questions can be addressed. So please type in also at the start of your question who your question is addressed to. So Nicole, Nicole and Sylvia, there is a question for you on will the new DAs or DAs with similar information sources, also called the Me Too, be expected to have data for all of the reference chemicals in the data set?
So I think that it's a great question. I think that's a question that needs to be taken up in the expert group on defined approaches for skin sensitization. I would say that that would not be a reasonable expectation. I would say that generating data and expecting the basis for comparison for for DAs with Me Too information sources to be 168 chemicals is not reasonable, and it's not consistent with any of the other OECD test guidelines. I could see, for example, because what we're really interested in is the comparison to the human reference data here, I could see like looking at the subset of chemicals that had both LLNA and human data, so that's 63 substances. I think that would be a much more reasonable sort of basis for comparison in terms of the Me Too DAs, or even just taking a carefully selected subset of those reference substances and deeming those performance standard chemicals the way other methods have a smaller subset of 20 or so performance standard chemicals in their guidelines. But again, it'll have to be discussed within the expert group. Sylvia, do you want to add anything to that?
No, I think that you have expanded on what was possible to say at this moment. It needs to be discussed in the context of the OECD work that will hopefully start on this. And it was a perfect summary.
Okay, thank you. We have a bunch of other questions that relate to the similar topic, the topic of mixtures testing. There were a number of participants today asking about the performance of the defined approaches for the testing of mixtures. Do you want to address that?
Sylvia, you want to go first this time?
Yeah, I can do that. Well, let's put it in this way. In the set of chemicals that were tested, some of the mixtures which were present were discarded, not because they were mixtures, but simply because there was not a reassurance that the same mixtures was tested in the in vitro methods and in the in vivo methods, and specifically in this case in the local lymph node assay. Though the individual test methods can be suitable for the testing of mixtures, apart from the DIPRA, where you need to have a defined structure tested there. And it is possible to test the mixtures, although, of course, I mean, we need to expand a little bit our experience with that, with the testing in general, with the individual test methods included in the DAs. So it's not a problem of the DAs, it's a problem of the individual test methods for which there is not so much, let's say, evidence on the testing of mixtures. But I guess that now, with the DAs being approved, there will be people starting testing on new chemistries, on new compositions, and including mixtures, and this will bring us much more insight on how well these methods perform.
Thanks, Sylvia. And I'll just expand on that to say that, technically speaking, within Guideline 497, it is clearly stated that that these DAs do not apply to mixtures per the guideline, right? But as I said, the NTP has been testing mixtures in the in vitro methods, applying defined approaches. We're analyzing that data right now. They seem to be working pretty well for certain types of mixtures. And I would say also that it's dependent on the regulatory authority that registrants are submitting the information to. So, I would always encourage you on industry side to communicate with the regulatory authority and see if they would be interested in, reviewing data on mixtures, because if it works for your substances, then it may still be accepted.
Thank you very much, Sylvia and Nicole, for these answers. And I invite you to maybe have a look at the Q&A box and type in some answers for other questions that were raised. We have a lot in there. So now we are almost on time. We are going to move to our next speaker. Thank you, Laura. The floor is yours.
Thank you, Anne. So just one moment. I will share my screen. And I hope you can see that now. Can you see the slide?
Yes, we do.
Excellent, thank you. So also good morning, good afternoon, good evening from my side as well. And thank you, Anne, for the opportunity to give this presentation here in this webinar. So my talk will be about on the use of defined approaches for skin sensitization under the REACH regulation, which is the European chemicals legislation. This is the outline of my presentation, where I will quickly go through the information requirement under REACH, the use of defined approaches for skin sensitization, our recently updated guidance on the use of defined approaches, some issues that we noted in the data already submitted to us, and some advice to the registrants that need to provide information under REACH. So why is information on skin sensitization so important? This is one of a common occupational and environmental health issue, and it is estimated that about 25 to 30% of industrial chemicals are skin sensitizers. And this is confirmed with the data that we have in our database. Therefore, it's really important to have reliable methods to predict skin sensitization potential of chemicals in order to protect the workers and the consumers. And this is one of the reasons why skin sensitization is an information requirement that applies to all companies that need to submit information under the REACH regulation. The legal text specifies the objective of the information requirement, which is that the information that is provided, it needs to allow the following. You need to get a conclusion whether your substance is a skin sensitizer or not. And in case the substance is a sensitizer, what is the skin sensitization potency? So whether the substance is a strong or extreme sensitizer versus a moderate sensitizer, meaning category 1A versus 1B, as indicated in the GHS or in the European CLP. Also, if your substance is a skin sensitizer, and then a risk assessment needs to be performed. If one is in the situation that the information that is already available doesn't allow to make these conclusions, and new testing needs to be generated, the testing shall start with in-chemical in vitro methods. The regulation doesn't specify which methods are to be used, but it requires that 3 key events of the adverse outcome pathway for skin sensitization are covered, which are the molecular interaction with proteins, inflammatory response in keratinocytes, and activation of dendritic cell. So this is the standard that needs to be provided. However, in some cases, you might have information, for example, from 2 key events that already allows you to make a conclusion on the classification and risk assessment. In such cases, the other key event does not need to be covered. And this is, for example, where the defined approach comes to play. As it was mentioned already in the previous presentation, that the available in chemical in vitro methods, they may not be suitable for all types of substances, or you might get results that do not allow to make a conclusion on the classification. So this could be, for example, you have inconsistent data and you don't know why, or you might get an inconclusive result from the defined approach. And then as a last resort, can an in vivo study be performed? And under REACH, if you need to resort to in vivo testing, the first choice of assay is the local lymph node assay. As I said earlier, if your substance is a skin sensitizer, you need to provide information on the potency. But there is one exception to the rule, and this is in case if you have an older in vivo study that was performed before the information requirement change came into force, and that was in May 2017. And that study doesn't allow you to make a potency estimation. You don't need to repeat that study. So this could be, for example, that you have an existing genetic study, and how that study has been designed is that you go as high as possible with the dosing, and because of that, you might not be able to conclude on the skin sensitization potency for that substance. So this was in brief, what is the information requirement for REACH for industrial chemicals. And I will go to the use of defined approaches under REACH. Just recently, we published an update of our guidance for skin sensation in chemical in vitro methods, and now it contains the use of the defined approaches. You can find that guidance on our website if you go to the support, testing methods and alternatives, and then you select the OECD and EU test guidelines. That page has the PDF version of this guidance, but it also has for other endpoints short guidances, like for irritation and mutagenicity and environmental toxicity. So what are the key aspects of the guidance? It lists all validated and OECD approved methods that are currently available and to what outcome they can contribute to. It explains how to use in chemical, in vitro, in silico methods, either within weight of evidence or by using a defined approach. It explains what type of outcome needs to be obtained. For example, you need to have the potency estimation for sensitizing substances. And it also lists the currently known scope and limitation of each available method and a defined approach. So going to our database, 2 years ago, we did an extensive data mining for the purposes of a report on the use of alternatives under REACH. And at that point of time, we found about 350 substances that had information solely from in chemical in vitro methods. And here you can see in the table what type of methods were provided in our registration database. And as you can see, we have a lot of information up from DPRA, keratinosense, and H-clad assay, indicating that at least for some of the substances, a defined approach could be applied retrospectively. There were, however, some issues that we noted when we looked a little bit more carefully into our database. For some of the substances, information on the required key events were provided, and classification was correctly considered, including the potency categorization. However, we had quite some substances where specific issues were not considered. For example, not all of the key events were covered, and it was not justified why 1 or 2 key events were missing from the information that needed to be provided. Sometimes we had also conflicting information, and it was not justified why a specific conclusion was reached. Also, we saw quite a lot of cases where the substance was predicted to be sensitizer, but no subcategorization was considered as required by law. So the registrant had selected category 1 as self-classification. Here is an overview of what type of approaches companies did provide to us when submitting in vitro in chemical data only. So here you can see in the table above that we have quite some substances where only 1 or 2 key events were covered, even though the standard is the 3 key events. One issue is good to note that especially the cases where we have information from only one key event is quite problematic, because currently, or at that time especially, none of the available methods can be used to make a standalone prediction on the skin sensation potential. The only alternative currently is the recently adopted kinetic DPRA assay, which could be potentially used as a standalone assay if the outcome is category 1A. In the table below, you can see what type of self-classifications were applied by the companies submitting in vitro in chemical data only. And as you can see, in some of the cases, potency subcategorization was considered, but there is a huge number of substances where the potency was not considered. So either that needs to be done based on the existing data, or perhaps additional data would need to be generated in order to be able to conclude on the potency. So to the advice that we have to our registrant is to get known with the OECD guideline and the guidance that we have prepared, to assess whether the defined approach could already be applied based on the data that they already have available and have submitted to ECHA. If a conclusive prediction could be obtained based on that data, it's advised to update the registration dossier accordingly. However, if one is in the situation that you get an inconclusive prediction from the defined approach, there might be a need to generate new data or to improve the weight of evidence assessment in order to get a conclusion on the hazardous properties of the substance. So at the end, I would just like to give a special thank to Andrea Gissi from ECHA, who was heavily involved in the defined approach project, especially with the QSR toolbox and in silico approaches in general. And I would also like to thank the OECD Secretariat and project colleagues Silvia Casati and Nicolas Kreinstreuer, especially on the good collaboration within this defined approach. Thank you. That was all from my side.
Thank you so much, Laura, for the presentation today. And again, we have some questions. We will take one or two. The first one, Laura, for you. For REACH, if new studies needs to be performed, is it mandatory to apply the defined approaches to perform those studies included in the guideline 497?
Okay, quick answer is no. One aspect is that, of course, if you do the defined approaches or you do the methods that are within the defined approaches, that gives you confidence on the prediction and also confidence for us when we assess that data. But one of the most important aspects is to consider whether those methods are the most suitable for your substance. So that should be the first priority when you select the best method for your substance. So I hope that clarified.
Thank you, Laura. And conscious of time, I think I will invite you, Laura, to have a look at the Q&A box and see if questions are addressed to you, and type an answer for our participants today. Thank you.
Okay, thank you.
Having heard from Laura on the situation in Europe under REACH, I would like to invite maybe our two panelists from the United States, Anna Lowitt and Paul Brown, to turn their cameras on and provide a little bit their perspective on these defined approaches and the implementation in your respective regulations.
Okay, I don't know, I guess I can go first if that's okay.
Yeah.
All right. So I am from the US FDA, and as Nicole already kind of showed a little bit of our perspective in her slides. I work mostly with human pharmaceuticals, so I can comment on those and the perspective we have on those. I mean, clearly with human pharmaceuticals, our interest is to identify clear positives before you go into the clinical trials, because you don't want to put patients or even healthy subjects that are in some of the clinical trials at risk of being exposed to a potent sensitizer. We're not necessarily focused on identifying, the weak sensitizers, because the great advantage, I guess you could say, with human pharmaceuticals is we will get the human data in clinical trials, certainly, including sometimes in provocative skin sensitization trials where you're specifically trying to see whether a drug is a sensitizer. And then we also will get post-marketing data once a drug's marketed, which, while it's not controlled, it's a fairly robust collection of data. So you will sometimes identify sensitizers in that setting. So again, our interest is mostly on actually not having too many false positives. So we can be satisfied with assays that might not have the highest sensitivity, but hopefully have a high specificity. And the reason for that is you don't really want to eliminate drugs that might be beneficial to patients, and you want to make sure that those can move forward. The human data, as I say, you'll get it from a couple different places, and that's, again, one of the great advantages from human pharmaceuticals. As far as guidance on skin sensitization, there are a couple different documents available. ICH has a guidance on immunotoxicity. It mentions skin sensitization, but it really doesn't give any detail. FDA does have guidance documents on immunotoxicity. One was finalized in 2002, so that's quite a while ago. It does have a fairly extensive section actually on type 4 hypersensitivity or skin sensitization, and it mentions several different approaches there in assessing it in non-clinical studies. Since that was quite a while ago, it really doesn't go into depth on the in vitro assays. However, it does actually mention covalent binding being used as a biomarker for skin sensitization. So it was sort of forward-looking and anticipating things like the DIPRA. Nicole mentioned in 2020, we published a draft guidance that included that comment on using a battery of alternative methods as a potential approach. The wording we put in there was intended, it preceded obviously the finalization of the defined approach guidance. But we wanted to try to include language that would capture that when it did become finalized. And so that's why the language doesn't specifically point to the OECD guideline, but it can certainly be interpreted that way, I think. And then since then, I've gone back and looked and talked to some of my colleagues that work with topically applied drug products. And in fact, we have received proposals from sponsors on submitting those in vitro assays, basically the battery. They even use language that sounds like the defined approach, but they don't refer to it. Some of those submissions, again, preceded the finalization of the defined approach. And in fact, we've gotten submissions with those data in it, where we've reviewed them and included it in our assessment. So I think going forward, if sponsors come in and talk to us before they come in with their actual application, we do have a process where we call it a pre-IND. So it's before you actually submit your IND to conduct clinical trials, where you can come in and propose, here's our approach, does that sound okay? And we can give feedback on whether it does or not. And we already have given that feedback on these in vitro assays and say, yes, that looks like an acceptable approach. I mean, some of the questions that have come up about looking at mixtures or final formulations, I mean, as Nicole said, the defined approach really says it's not for mixtures. And I think that makes sense if you think about it. I mean, some of these in vitro assays, you really can't put an ointment or a cream on the cells directly, so you can't really assess that final formulation for a human drug product. However, when most of the ingredients or all the ingredients are commonly used in drug products already, and there's really maybe only one new ingredient, the new active ingredient, then that's a situation where that one active ingredient could be assessed, I think, in these alternative approaches, and that would presumably be a sufficient evaluation. So it really will depend a little bit on the specific situation, maybe even the indication that it's being used for, as far as whether it be an acceptable approach, which is why I think it would be wise for anyone considering it to talk to the agency before they initiate it. So I think I'll stop there so I don't take up too much time, and we can get to other questions. And so Anna can weigh in.
Yes, please, Anna, go ahead, and there will be maybe time for 1 or 2 questions to each of you in the end. Thank you.
Okay. Hello, everyone. Thanks, Anne, and thanks, Paul. So I don't have a lot to say because I think between Sylvia and Nicole and Paul and Laura, so much has been said. It's nice to go last, actually, or near last. So for those of you who don't know me, my name is Anna Lowett. I'm a senior science advisor at EPA's Office of Assay Programs, which is part of the Office of Chemical Safety and Pollution Prevention, for which my sister office, the Toxics Office, together we regulate pesticides and toxics in the United States under multiple statutes, notably the amendment to the TSCA Act, the Lautenberg Act, that provided for toxic chemicals the legal need to evaluate the existence of alternative data before asking for in vivo data. So unlike some regions in the world, in the pesticide space, we don't have any legal requirements for reducing animal use or moving to in vitro. But both our toxics office and our pesticide office have been working in the space for more than 10 or 15 years now. Not only because it's just the right thing to do as good public servants, but because we believe that moving away from many of the whole animal tests to the in vitro assays are actually more human relevant. And we're finding that to be the case, certainly with the skin sensitization, like you heard from Sylvia and Nicole earlier, that the in vitro assays actually do a better job of predicting the human response than does the LLNA. And that's super exciting, but it may not always play out that way, but certainly has in this case. So we have had on the books now for several years what we call a draft interim science policy, allowing for the submission of 2 defined approaches, one of which is now in the DAS guideline. Both our toxics office and our pesticide office, we don't need a guideline for potency categorization, but we do need evaluation of that sort of up-down yes-no answer. And in certain isolated cases, as with our isothiazolone assessment, we do need to be able to quantify skin sensitization potential for risk assessment purposes. So we've been asked multiple times in the last 6 months, are we going to be updating our guidance? And I think we haven't quite figured that answer out yet. But as the DAS is now part of mutual acceptance of data, we will accept those data under MAD. So to the extent to which maybe you're in a company and thinking about submitting things and haven't seen that draft guidance go final, I think you can, with full confidence, begin to submit those data. I did do a count last week of how many had been submitted to our 2 offices, and the numbers are definitely smaller than in the REACH program. And at ECHA, our toxics office has in the order of about 50 or so, and our pesticide office, we're still under 15. So even though we've had a policy on the books for several years now, the momentum is still building on companies making the transition from the animal study to the in vitro. And the hope is that with the passage of the DAS guideline back in April, or in the last few months, that momentum starts to substantively build. And similar to Paul and Laura, we would really highly recommend that companies come to us in what we call pre-registration meetings or early registration review meetings and talk to us about submitting the in vitro assays, as they're not only avoiding the use of the animals, but they are just more human relevant and give us so much better information. So just a couple of things, like many of you in the Q's and A's and some of the comments, we are keenly interested in the work on the mixtures. And in fact, the pesticide and our toxics office both have been working closely with the National Toxicology Program as part of that effort Nicole talked about to evaluate the extent to which the in vitro assays can work for different types of mixtures. And having seen some of the early preliminary data, some of it's really exciting and some of it's a little, I don't want to say disappointing, but it's definitely, there's a lot of work there to do. There's still work to do. In my personal view, I don't think we're imminently about to change from just single agents to formulations, although some specific sectors are definitely closer than others. So there's definitely a lot of work there to do. And I think it's an area where the international community can probably come together, maybe a little bit better and come up with a strategy of how to do that. So I would encourage people who are interested in helping out with that work to come forward and, and talk to the technical leads and others who have interest in this area. So maybe we get a better strategy on handling the mixtures issue. Excuse me. And one other thing I wanted to highlight is that, as Sylvia brought up, we've had a really unique situation with skin sensitization that there's a lot of human data. And in the drug space, as you heard from Paul, as things will move into human clinical trials, there will be human data. But in the industrial chemical and the pesticide space, the number of chemicals with human data is very, very, very small in relation to the totality of the space. And so I really want to make sure that we're not holding new guidelines up to that existence of human data for environmental chemicals, either in the industrial chemical space, the toxics, or in the agrochemical or biocide space, where the data aren't going to be there. And so we need to think about how to do these evaluations and these confidence building exercises where there is limited or no human data. And that's going to be a challenge. And I know a lot of smart people are working on that. Those are the points I wanted to make. So I appreciate being included and looking forward to the questions.
Thank you, Anna and Paul, for your comprehensive interventions. Maybe we take one question for Paul. Is the FDA accepting DAs-based data for skin sensitization for medical devices? Could you address that question, Paul?
Unfortunately, I'm not in the Center for Device Evaluation and Research, so they'd be the folks that would be able to comment on that. I know they're aware of the approaches, and I think they are considering whether they'd be acceptable. I'm not sure if all the types of chemicals that they usually have to deal with have been addressed in all the validation data. I'm not entirely familiar with that, but I know they are aware of it, and I think they are considering it as a possibility.
Okay, thank you. Thank you very much. I think the rest of your interventions have addressed a lot of the questions posed in the Q&A box. So we are going to move now to our next and last presenter for today, André Mueller, who is going to talk about the activities under the United Nations globally harmonized system for classification and labeling. So André, floor is yours. Thank you.
Yeah, hello everybody, and thank you, Anne, for the possibility to present on our work ongoing at the DHS. In the coming 10 minutes, I want to provide you a short intro to the DHS and how we work there. What have we done so far on non-animal test methods and ongoing work on the inclusion of non-test methods for skin sensitization. Now, first, what is DHS? DHS is the globally harmonized system for classification and labeling of chemicals. And with chemicals, we mean substances and mixtures. And the goal is to have the same information on the label of every substance and chemical overall over the world. So it's an agreement between countries to apply the same criteria, labeling, safety data sheets everywhere. And those agreements are included in the DHS, which is in practice the purple book you see here. And then countries can include the DHS in the national legislation using a building block principle, which means that you can choose which type of effect you want to include in which hazard classes. You don't have to use them all. The DHS is already applied in approximately 80 countries, and more countries are working on it. And then the DHS is not static, which is dynamic. So we update it every 2 years, and it is done by a subcommittee. and the formal meetings takes place at the United Nations in Geneva. So we have a 2-year work program, and all updates require consensus by the member states. Now, the actual work is done by informal working group, and those groups consist of participants from member states, industry, organizations, and NGOs. The work that can be done in such a working group is determined by the mandate agreed by the subcommittee. And at the moment, UK and the Netherlands, with support of the GRC, leads a non-animal testing informal working group that has a mandate to amend the criteria using non-animal methods. We will take a closer look at that mandate. And then we can identify available in vitro and in chemical methods validated at an international level that could be used, identify existing guidance for such approaches, assess whether substances and mixes can be classified using those non-animal methods, and then assess whether how to combine such information, just whether in an integrated approach, in a tiered approach. And if there's agreement within the working group, then we prepare draft amendments and additions to the JHS that can be used for classification, and all that is discussed within the informal working group. And up to now, when there was consensus in the working group, then there was not much discussion in the formal subcommittee. So what have we done previously? The non-animal test method working group has worked on skin and eye irritation. For skin corrosion, which is chapter 3.2 of the GHS, we have included in vitro methods and non-test methods, which includes things like QSARs and read across, and they were placed in revision 8 of the GHS. For eye irritation and serious eye damage, we introduced in vitro methods and non-test methods, but also defined approaches, because for eye irritation and serious eye damage, there is already an OECD project going on, which includes defined approaches. Then we also allowed such in vitro methods and also non-test methods to be conclusive also for negative outcomes. And this replaced a single statement that validated and accepted in vitro methods could be used, and the same for things like read across. So the criteria are now much more in balance, where previously we had a whole page of describing the criteria for animals and 1 sentence for in vitro methods. Now it's much more balanced with about 1 page for both. So what is the work where we have to do when we look at skin sensitization? Now we have criteria for classification based on human or animal data. And if those criteria are not met, then the remaining option is a combination of 2 more indicators of skin sensitization. And among those indicators is number D, positive data from non-standard methods, and E, positive results from close structural analogs. When those criteria were developed, we hadn't any validated in vitro or in chemical tests or defined approaches. So it's unclear whether option D would include such methods. Secondly, they couldn't be used as standalone, because you always had to combine these 2 types of such indicators to come to a classification, to come to a conclusion. However, now we have validated and adopted in vitro methods and defined approaches. So there was a clear need to amend the criteria for skin sensitization and introduce these into GHS. So what is the current status of those discussions? We started with a paper developed by JRC describing the main issues, and it was inclusion of OECD accepted in vitro and in chemical methods that could be used as standalone for category 1, meaning not for no classification. inclusion of the OECD accepted defined approaches for category 1 or 1A and 1B as standalone criteria. Inclusion of non-OECD accepted in vitro and chemical methods is still under discussion, and the same applies for non-OECD accepted defined approaches. Also, the non-test methods, so the read-across, can be introduced as standalone criterion. And then the ongoing discussion is mainly on how to combine those information if they are available. Do we then do it in tiers or a weight of evidence approach? And that is still the discussion. We have not yet drafted any text. So the discussion is still going. Next week we will have even this week we will have a discussion regarding a tiered approach. We have made a first suggestion, and it's the first tier that includes human data, animal data, and the result of the defined approaches. If those results are conflicting, then we apply a weight of evidence within this tier. If this tier is not conclusive, we go to a second tier, which include the individual in vitro and chemical methods that can be used to classify as a category 1 if they are positive. And we may have to split this in case of mixtures. And we can use non-test methods like QSARs and read-across. If this is also not conclusive, we have a tiered tier that is comparable with the current second tier, which includes a number of indicators. And finally, the proposal is to have an overweight of evidence in place. All those previous tier are inconclusive. So that was it, and I'm happy to answer any questions.
Thank you so much, André, for this interesting and comprehensive presentation of the UNGHS activities underway. There is one question for you. So the OECD also looked at several other defined approaches for skin sensitization, but only 3 are currently included in Guideline 497. The question is whether these other defined approaches that will in future be evaluated, as we heard from Nicole, can they also be used for classification?
That's still ongoing. When looking at the previous endpoints we had, skin irritation and eye irritation, then the solution to that was that we give the individual countries the option to decide how to use it, because that's often more easily resolved than waiting for an update at the GHS.
Thank you, André. And we are reaching the end of our webinar today. I would like to thank all of the panelists today for their time preparing for their presentations and interventions. I hope all participants found useful information in this webinar. I remind you that the recording of the webinar will be available soon from our OECD public website and also the slide presentations from the speakers today. So again, thank you to all of you, and I wish you all a nice day. Thank you. Bye.